There was one experiment my first semester as a graduate student, and it was just counting individual muscle fibers, single muscle fibers. And I would do hundreds and hundreds of embryos. So sort of that repetition over and over and over and over again, you're no longer thinking that you're looking at muscle fibers. You're able to appreciate different qualities of each fiber and that repetitive nature of looking at each individual component. This sort of freeing from thinking of the whole structure itself and appreciating the quality of each of the components. All right, so I'm Frank Stellabotte, and I work in a lab that studies vertebrate muscle development in Stephen Devoto's lab at Wesleyan University. The research I do here now in the lab is I study how embryonic muscle grows to its adult form. And my research specifically targeted the cells that are responsible for growth of the muscle from an embryonic form to an adult form.
The animal that we work on is the zebrafish. The zebrafish embryo is optically transparent, so we can focus the microscope through the embryo and watch development happening in real time on the microscope. And so we can see patterns emerge in the embryo as they're happening.
This is an image from my current research. So this is a zebrafish embryo. I've labeled each cell boundary with a label called phalloidin, which picks up actin that collects, protein that collects at the cell boundaries. And then in red here, you can make out individual nuclei that I've labeled with an antibody that recognizes cells that are about to become muscle fiber cells. And so when I labeled like that, I could see that some cells are going to become muscle fiber cells right away, and then other cells don't express that, and they remain as a population that gives rise to growth of the embryo. The techniques I learned as a painter inform what colors I choose to label individual cells in the embryo, because once you've taken an image, it's usually a black and white image, and then you pseudo-color it. But what colors you choose, that decision is greatly informed by my experience as an oil painter.
Frank's skills as an artist, I think, play very closely into his skills as a scientist.
I actually don't even remember not having art being a part of my life. I've been really lucky because most of my advisors that I conducted scientific research with sort of fostered that artistic side of me and the creative side of me. It was really interesting, but the artists I work with, they can sort of sense that I'm a little bit different than the other people in the room, and the way I pursue things and the directions I take and what I think is important. Yeah, so a general theme I have, that kind of sort of a thread that flows through all my paintings, is this idea where you take an object that's something very recognizable, break it apart into pieces that abstracts the piece. My goal is sort of to free the viewer from the constraints of having a name associated with what they're seeing, liberating the person from all the preconceived notions about that particular object, and sort of appreciate the object from a different viewpoint, sort of appreciate each aspects. And here's another painting where I was sort of inspired by Chuck Close in his later work, where he took pieces of color and sort of like each individual piece of color, like each component is sort of like its own little painting, and then they're sort of put together to create an image. And I always really appreciated that, because you could appreciate each individual component at a smaller scale as if it were its own painting, and then combine it all together to create the entire image.
And his experience in creating these structures, these paintings or other structures, I think motivates him to understand how the embryo can create structures, create form out of a previously blank slate.
So when you look at an embryo, an individual cell of an embryo in the microscope, you sort of have a feel for the complexity of that cell, and you see so many processes and so much complexity going on there, and you have a greater appreciation for each individual cell in the living embryo. Once you have that appreciation for each component of the embryo, and if you think of all the things around us that are made up of so many different cells, sort of have a greater appreciation for life in general. That's the message I try to convey in the paintings that I create, and also the images I create using the microscope. The hand-drawn images in the paper we just published, I think, are very important. The drawing sort of takes on a sort of classical style that isn't common in science anymore. Scientists would look at a section in a microscope and draw every single form, and they would use watercolors or other media to draw the cell types. So in this most recent paper, I sort of took that same style and applied it to sections I took of the zebrafish embryo. After I leave Wesleyan, and I defend my dissertation, I'm going to be going to the University of Southern California at the medical center.
The visual artists that are in Los Angeles, I think, have a dramatic impact on the way science is done. And I think Frank will fit in very nicely there. I think his artistic flamboyance and creativity will be celebrated there. And I think he will both be inspired and inspire the scientists who are working in his new lab at USC.
Blank Frank is the messenger of your doom and your destruction.
Bovine serum